Oh Lord, it's blackzonda. Oh Lord, it's blackzonda. Glitch Crusade. Fucking HanSolo did his shit. Okay, let's use some brain.
Boss.
In the normal transmission of pain, ascending nociceptive signals travel up the spinal thalamic pathway to the thalamus, where they are processed and relayed to cortical and other areas. The ascending pathway also relates to the periaqueductal gray matter, an important part of the descending pain modulating system, where specific cannabinoid receptors are located.
Cannabinoid receptors.
Stimulation of the periaqueductal gray is known to produce an analgesic effect. However, this neuroregulatory process is thought to be tonically restricted by inhibitory GABAergic alpha cells. Ascending pain signals relayed through the periaqueductal gray suppress the activity of GABAergic alpha cells through cannabinoid modulation, thereby increasing activity in the descending pain modulation pathway and increasing the analgesic effect. This is called depolarization-induced suppression of inhibition. A synaptic feedback mechanism is believed to be an inherent part of our normal neuroregulatory processes, including descending pain modulation. In many regions of the CNS, including the periaqueductal gray, the generation of postsynaptic action potentials is modified by an endogenous cannabinoid regulatory system. Endogenous cannabinoids are synthesized on demand from the phospholipid bilayer of the postsynaptic membrane. These lipophilic ligands are released directly into the synaptic cleft and act in retrograde fashion on the presynaptic neuron where the cannabinoid receptors are expressed. Binding of the endogenous cannabinoids affects intracellular signal transduction pathways, reducing the influx of calcium ions into the presynaptic neuron, causing a decrease in further neurotransmitter release. This in turn influences the frequency of postsynaptic firing. The administration of exogenous cannabinoids orally or through inhalation may therefore provide an option for treating pain that has responded poorly to conventional pain therapy. When exogenous cannabinoids are administered orally, they travel via the GI tract to the liver, where they are metabolized before entering the bloodstream and then the CNS. The onset of action from oral administration is less rapid compared to the inhaled route. When exogenous cannabinoids are inhaled, they travel through the respiratory tract to the alveoli of the lung, where they diffuse rapidly into the circulatory system. They then travel through the bloodstream towards the CNS, where they diffuse across the blood-brain barrier and into the surrounding tissues of the brain and spinal cord. Exogenous cannabinoids exert their modulatory effects upon the abundant cannabinoid receptors distributed throughout the CNS. The effects of cannabinoids may be illustrated by comparing three neuronal synapses showing normal pain transmission, the influence of endogenous cannabinoids, and the influence of exogenous cannabinoid administration. Exogenous cannabinoids from the bloodstream bind to cannabinoid receptors on the presynaptic neuron and mimic effect by decreasing influx of calcium ions. In this way, pain and other neural processes are modulated. The clinical effects of exogenous cannabinoid administration are not limited to pain and have proven effects including the reduction of anxiety, spasticity, anorexia, and nausea. The development of novel drugs to modulate endogenous cannabinoid reuptake and metabolism may also prove beneficial in pain therapy. The therapeutic benefits of cannabinoids, including modification of the endogenous cannabinoid signaling system, should be further explored through clinical research.
Despite all the difficulties, there's a group of doctors in the U.S. and abroad who have been advancing the research on the medical use of cannabis with highly impressive and sometimes revolutionary discoveries.
In a scholarly Cancer Review article published in 2009, there were over 421 chemical compounds identified in the cannabis plant. And within this group of natural substances, there are many chemicals that have significant anti-tumor properties at low enough doses to where they would be effective cancer treatments. That's an important part of this. It's one thing to be able to kill a cancer cell, but could you really survive such a treatment?
A botanist from the University of Colorado Springs branch, Robert Melamede, who has written an article sort of comparing the carcinogenic effect of cannabis with tobacco and explaining why he thinks that cannabis has the anti-cancer effect for lung cancer.
There's nothing else that impacts on so many of our illnesses. You see, when I talk about these age-related illnesses, we're all aging, but we don't all come down with cardiovascular disease. We don't all come down with autoimmune disease. We don't all come down with cognitive dysfunction. We don't all come down with cancer. But the chances are we're going to come down with one of them. All right? And here we have this one drug that's able to help us holistically with our biochemistry to restore balance. You know, cannabinoids kill cancer cells in many cases. People are not aware of that. They think cancer, cannabis, anti-nausea. It's way beyond that.
There are also studies taking a look at other organ systems that have found that marijuana seeks out the cancerous cells and preferentially kills them over the healthy cells.
There's a wealth of laboratory evidence that these anti-tumor properties kill cancer cells in a variety of ways. There are multiple mechanisms of action identified by which cannabis kills cancer cells, and they're divided into various categories. And among these are anti-proliferative effects. Normally, that's one of the hallmarks of a cancer cell, is that it just keeps reproducing. So if you stop the reproduction, that's an anti-proliferative effect. There are anti-angiogenesis effects, and this means that the cannabinoids will stop the tumor from being able to elaborate or grow new blood vessels to support the growth of the tumor. There are anti-metastatic effects, and that is simple enough to mean that the cannabinoids block the ability of the cancer cells to spread into other tissues. And there's another effect that has a wild name, apoptotic effect. Apoptosis refers to the ability of cannabinoids to speed the death of the abnormal cells. That's something that is especially important in cancer, because you're able to hasten the death of the cell without disturbing the normal cells around it.
Seth Research Laboratories in California have recently demonstrated that in some tumors, cancer cells are killed by marijuana, while the other healthy cells are left untouched. Cells that stop moving and become still white dots are dead cancer cells. The ability of cannabinoids to kill bad cells while protecting healthy ones is particularly important when we're talking about brain cancer because of the so-called blood-brain barrier. The brain has to be sheltered from outside influences that might hitch a ride on the bloodstream and cause havoc. What is exciting and unique about cannabinoids is that they can pass through the blood-brain barrier because of their slippery, fat-loving nature. Cannabinoids get right into the brain's cancer cells by moving easily through the cell's membranes, which are also composed of lipids.
As the primary active constituent of the medicinal plant cannabis, also known as marijuana, most people are familiar with the euphoric effects of this compound. After entering the bloodstream, this compound becomes available to individual cells in the brain. Delta-9 THC interacts with specific cell surface receptors. It is analogous to a key entering a lock. This triggers a reaction inside the cell. Unexpectedly, this reaction in normal cells is significantly different compared to the reaction in cancer cells. In cancer cells, these reactions lead to cell death. In normal cells, it doesn't. This surprising difference is likely due to two things: a greater number of receptors on the outside of the cancer cells, and a different type of reaction inside the cancer cells as compared to the normal cells.
Get it?
So what we found, a major finding, is that if you combine CBD with THC, you actually enhance the anti-cancer effects of THC. It's been previously reported that THC was effective at inhibiting aggressive brain cancers in vivo, and this, what they call preclinical data, led to a small clinical trial in Spain. So there's been quite a bit of literature suggesting that if you combine cannabinoids, actually different components of cannabinoids, THC and other similar compounds of similar structure, you might be able to enhance the activity of that individual compound. So this is the hypothesis that we worked with. We screened a variety of cannabinoids and against many different types of cancers. And from that screen, we found that this compound called cannabidiol, which happens to be the second most abundant cannabinoid in the plant cannabis, was actually the most active compound when we were testing its ability to inhibit many different types of aggressive cancers. So we combined this compound along with THC in culture dishes of aggressive brain cancer cells. And what we found is we got a synergistic improvement in the ability of THC to not only inhibit the growth of these aggressive brain cancer cells, but also to cause programmed cell death, which is very important because you actually want to kill these tumor cells so they don't have the ability to grow back. So that's really the major finding. And also within that, we were able to find specific molecular mechanisms that might explain why combining the two compounds makes for a more efficient treatment. And so we're hoping maybe to use that finding to potentially improve treatments, not only in aggressive brain cancer, perhaps in other aggressive cancers.
The evidence is piling up in mice-infested labs that the endocannabinoid system, when stimulated by cannabinoids, has an anti-tumor effect and can instruct cancer cells to commit suicide.
This was done by Manuel Guzman's group within the past less than 10 years, and what they showed there was that originally that THC, when injected into a brain tumor in mice and rats, a significant number of those animals the tumor would regress and disappear, so that you actually had survival of rats that would otherwise die. And they examined all the surrounding nerve tissue, and that was all fine, because remember, once again, cannabinoids protect nerves.
Dr. Manuel Guzman is a professor of biochemistry and molecular biology in Madrid, Spain, and is known for his groundbreaking studies on medical cannabis.
Hemos observado que los cannabinoides tienen un efecto inductor de la muerte de las células tumorales, un efecto inhibidor de lo que es el crecimiento, la multiplicación de las células tumorales. Lo que hacen es disminuir el crecimiento de los tumores.
If cannabis might be the miracle cancer cure that everybody's been searching for, then why don't doctors everywhere know about it?
People have a hard time believing that cannabis can have all of these fantastic effects that are described. But what we're doing is we're just stimulating a natural system that's already there. This has been developing for hundreds of millions of years. The invertebrates, the sea squirts, the hydra, there are primitive endocannabinoid systems in those organisms dating back 600-700 million years ago. The cannabis plant came along maybe 50 or 60 million years ago.
Cannabis is a natural substance and therefore can't be patented. Anyone with a green thumb can grow it, and so its production could be in the hands of the people, not the corporations that run a $200 billion a year cancer business. It may not be a coincidence that pharmaceutical companies are silently preparing for what could easily become a total revolution in the curing habits of the world's population. GW Pharmaceuticals, a company that specializes in the study and commercialization of cannabinoids, has already established a full-cycle facility in an undisclosed location in the south of England, where they oversee the cultivation of the plant right to the finished medical product.
Growing areas?
This facility grows 30 to 60,000 plants every year. Once the marijuana is ready for processing, it undergoes the same exact treatment for extraction and refinement that is forbidden to regular citizens around the world.
That looks some good weed. We don't get that. Look at that juice there. He's got the resin and everything.
Amazingly,
Damn.
GW Pharmaceuticals, who is this company that has 6 greenhouses in a secret location in the south of England.
GW Pharmaceuticals in the south of
Each
England.
greenhouse has a different strain of marijuana in it. Each greenhouse has 10,000 plants in it. Each of the plants is a clone of the other. So they've standardized this stuff. Now, their product was approved for sale in Canada for treatment of multiple sclerosis, the muscle spasm and the pain related to that.
And this is a spray.
This is an under-the-tongue spray. And the reason they went with the spray was they felt that they would have a hard time getting by the regulators if they had people smoke it.
In the United States, things are moving fast as well.
Breast cancer kills more than 41,000 American women every year. Researchers in the San Francisco lab are working to shrink that number, and they're doing it with an unusual weapon, marijuana, or at least a compound from it.
And what we found is that this compound called cannabidiol was particularly effective at inhibiting aggressive breast cancers.
Intriguing answers started appearing in the early 90s when researchers pinpointed receptors in the brain and the body that bind with the cannabinoids. Receptors can be described as locks on the surface of a cell, and when the correct key binds with the correct lock or receptor, it opens the door and delivers messages. Sometimes the message is that the body is feeling pain. Other times the message may be that there's an invader and the immune system must attack.
Scientists located 2 receptors, cannabinoid receptors, one called the CB1 receptor, mainly in the brain, and the other is the CB2 receptor, which is mainly in cells of the immune system.
The CB1 receptors are extremely abundant in the brain, but they're also found all over the body